Hi, my name is Matthew Yang, and I'm a junior at Centennial High School in Ellicott City, Maryland. For the Congressional App Challenge, I created CellposeCellCounter, an app, as you might have guessed, for biologists. And so what CellposeCellCounter does is that it uses AI models to automatically detect and count cells using a process called segmentation. Built on the CellposeSAM architecture, it helps researchers obtain crucial measurements that normally would take hours, reduced to mere seconds. CellposeCellCounter provides an AI-powered solution. I trained custom CellPose models in the CellPose GUI for increased segmentation accuracy. These models were uploaded to a GPU-accelerated Hugging Face space where a GUI was created with Gradio using Python. Here's a bit of the code I wrote for the app. I have functions to run segmentation, calculate metrics such as confluency and viability percentage, and options to process images after uploading. Now let's see it in action. So let's say you want this sample of a hemocytometer and you want to know the cell count in it. What we can do is upload the image, select our model, which in this case we'll use hemocytometer model, and run segmentation. Looks like it's predicted 6 seconds for inference time, and we'll see, yeah, around 6 seconds, and we already have our cell count. 55 cells counted in less than 7 seconds, and much faster than a human could count. You can see all of our cells are accurately segmented, and we also have another interesting feature I've developed called viability assessment, and I'll run segmentation on this sample so we can have a clear example. We have 67 cells, and we can go here. So viability assessment is basically a way for us to determine if cells are alive or dead based on if these cells take up a dye called trypsin blue. These darker blue stained cells are dead, and they've taken up this dye because their membranes are compromised. And so we can use this metric to determine whether cells are alive or dead in a kind of thresholding tool here that I've developed. And finally, let's say you don't have something in a hemocytometer. You have, a kind of like this. This is a photo of glioblastoma tumor cells. Glioblastoma is a type of brain cancer, and one of the main reasons why I wanted to develop this app was to use this app to analyze growth of tumor cells over time. And so for this, obviously not the same kind of cell shape as this, we'll use the general model. I'll run segmentation one more time. We should get a cell count. Takes a bit longer. And yes, so we also have segmentation on different kinds of cells. We can also have this confluency measurement, which is used for these larger cells, which is the percentage of space that the cells take up. And so CellposeCellCounter can generalize to different kinds of cell types as well as provide accurate measurements for all of them.